Oh, okay. Couple of things with the greenhouse. I believe it was last Thursday or Friday. They went in there and turned off the swamp cooler system. So the swamp cooler is what does the majority of the cooling in there, but they have to shut it down when the nighttime temperatures get close to freezing or start freezing temperatures because the water in the pipes will freeze. So if you have plants in there currently, especially on a sunny day, today is probably the only day this week we need to worry about it so much. The temps could get quite warm in there and there's no swamp cooler to kick on. So the only way to cool currently is with opening up the exhaust fan, turning those on and opening up the roof vent is the primary cooling mechanism. So keep that in mind if you have plants in there. And then the other thing too, if you need to keep your plants going over break, talk to Duncan about that. And we'll make sure that things are taken care of if you can't, someone in your group is unable to do that. So talk to Duncan and we'll figure something out. Okay. Okay. With this mechanisms of defense, I think we left off here looking at the idea that once the plant gets the signal from, in this case, we're looking at microbes primarily, so bacteria and fungi. Once they get that elicitor, something coming from the pathogen, and that docks into the appropriate receptor, what they're trying to show here is that you have a series of events that can occur, including that signal transduction process, which they're alluding to here. And so you can also see that there's calcium in play here as a second messenger. And so you can see calcium is coming through into the cell and starting to act as a second messenger. And then in this case too, this is relatively new information where they're finding out how calcium is influencing nitric oxide production, which is actually a good thing. Okay, that's part of their ability to defend themselves. So we'll talk a little bit about that. One of the things that they can do when they are picking up that something's attacking them is this reactive oxygen species, which we've talked a little bit about already. But here you can see that you don't need to know the details of how it's made or the enzyme, but basically you get the formation of reactive oxygen species. And this is happening here. I might have showed this already, where the idea of when oxygen starts to accept electrons one by one by one, you can get these intermediates that are reactive oxygen species. And so this just happens naturally. And in this case, they can ramp it up and deliberately do the production. In this production here, what this is showing is as oxygen is being reduced all the way to water, this is primarily happening in the mitochondria. It's happening right in your mitochondria right now. It's happening in the plant's mitochondria. But you get these reactive oxygen species, free radicals that form, and hopefully they're brief and they'll go to completion where they don't persist and you'll get water as a result. But every once in a while, you can get issues where these build up and start to cause problems. So it can be a bad thing. Here, though, it's a good thing in the sense that sometimes reactive oxygen species are made in direct response to the pathogen, and it helps protect the plant against that particular pathogen. So you've seen this already as well. And so the reactive oxygen species, they're starting to map this out more and more with plants, and they're finding that it really is a common tool that they use no matter what's facing them, whether it's an abiotic process that's stressing them out, like too much salt or too cold or too too little water. but they're also showing it in response to biotic attack by pathogens too. And so in this case, yes, it can slow down their growth, but it's actually a good thing too, because it's helping them respond appropriately to whatever stress is that they're experiencing. So one of the things that can happen, for example, is, let's go back to this first. So as that reactive oxygen species is kicking up, hopefully that's taking care of some of the issue. But you can see that sometimes that will act as a signal cascade and can turn on other responses. And so you see the white boxes there that there's several different ideas. So I want to go through some of those. So with the hypersensitive response, that was probably one of the first things discovered in terms of how a plant is defending itself against a pathogen. And primarily this, I think the early studies were done with fungi attacking the plant. And what they show is that as the fungus starts to penetrate into, let's say, the leaf, and the leaf cells are getting exposed to the fungus, the cells that are right around the fungus start to die. And it's a very deliberate thing. It's from the plant's end of it. So the plant will deliberately tell those cells to die. And so the fancy word for that is apoptosis. But essentially, you'll see in the process, the fungus invades, the cells around the fungus start to die quite rapidly. And the idea here is that as those cells are dying, the plant can kind of suck away the nutrients away from the fungus so that the fungus is kind of surrounded by like a desert of low nutrients or no nutrients. And that's the goal, is to starve out the fungus. And so what happens is the pathogen starts to slow down its growth or invasion process because it's not finding any nutrients. So it's starting to get nutrient deprived. And when they notice this, what they saw is often it will have this burst, this what they call oxidative burst, right before this process happens. And again, it's that reactive oxygen species. And so they started linking this idea that, oh, okay, first there's that reactive oxygen species that kicks up, and then that triggers this hypersensitive response. And it seems to, there's several different flavors of reactive oxygen species depending on the plant that they'll be paying attention to, and then the hypersensitive response kicks in. And so nitric oxide is also another process that kicks in. And so here, it's a second messenger. They used to be called secondary messengers, so sometimes I accidentally type that, but current verbiage is second messenger. And what they're finding is that the plant needs both reactive oxygen species and the nitric oxide together in order to really hammer home the point. You need to start kicking up your hypersensitive response. So if we go back to this diagram, you can see here the nitric oxide. Okay, they're showing the nitric oxide production, and then they're trying to show that it's acting as a second messenger. Same with the reactive oxygen species. And then you see the hypersensitive response. Okay, so they're starting to see that you have to have this kind of 1, 2, 3 punch in order to get the hypersensitive response kicking in. Whoops, not that. What happened there? That was weird. Let's go back to that. Okay, so this is kind of the hypersensitive response is kind of a mixture of responses, because you have the local cells starting to die and then the nutrients are getting sucked away. And then here's some results from one of the experiments that they did where I don't know if you can see the fungus from where you're sitting, but there's some fungal hyphae in here. But the left image is when the plant is fully up and running with the whole hypersensitive response. And you can see the fungus didn't get very far. It's not spreading very far. Whereas the one on the right here, this is, you know, quite successful infiltration of the fungal hyphae as it progresses through all the cells in that area. So the other thing that tends to happen now, if starts to be successful and you have that stalling out of the fungal growth, another thing that starts to happen right around where the fungus is, excuse me, is this ramped up lignin biosynthesis. So if you can imagine, you know, right around the fungus that's trying to get access to all those nutrients, all those cells have died and the nutrients sucked away. And then the plant starts to like build a wall around that area. And so the lignin biosynthesis right in that area starts to ramp up. And it's like creating, you know, a big brick wall around the fungus. Depending on where it might be callus in the phloem tissue. It's not a big deal, but just realize there's slightly different strategies depending on where the fungus is. But generally, you can say the plant starts to ramp up its physical barriers around that fungus. And hopefully the fungus is on its way to dying because the nutrients are getting sucked away from it. And then kind of just walling it off. So if it's gets reactivated somehow, hopefully it won't make it past the physical barriers. Now, let's say maybe that didn't work so well and some of the fungus is still living, or maybe a new fungus comes in while the plant's trying to deal with this fungus. And the fungus starts to gain an upper hand. Then the plant can start to invest more direct resources by creating enzymes that specifically attack that pathogen. And so these enzymes they call them pathogenesis related proteins. That's the name of them. So there's a bunch of different enzymes. And so they just call that whole group of enzymes pathogenesis related proteins. So they're related to the pathogen, that whatever pathogen is attacking them. And so usually they're enzymes that attack the cell wall. So fungi have cell walls, bacteria have cell walls, different types of cell walls, but they do. And so the plant can produce different enzymes to attack those cell walls. So glucanases, chitinases, hydrolases can start to break down the cell walls. And if you do that, the fungus and the bacteria tend to die. And so because there's so many different enzymes, they just lump them under this category called pathogenesis related proteins, PR proteins. So if the hypersensitive response doesn't work, the building a physical barrier is not working so well, then they'll start to do a more direct attack on the actual pathogen. And in this case, one of the first things they'll do is start making those enzymes, and hopefully it breaks down the physical cells of the pathogen. So that's where they're getting this in the white box. It says biosynthesis of hydrolytic enzymes. That's the pathogenesis related proteins. Another discovery that they made, which is being shown here, is phytoalexin biosynthesis. Phytoalexin biosynthesis. So these are molecules, and again, this is like an umbrella term because there's lots of different phytoalexins. It depends on the plant species. But one thing they share in common is that they're really strong inhibitors. There's like potent, almost like antibiotics, if you want to think of them that way. That's a good analogy. And like I said, there's a whole bunch of them out there that they've discovered. And so they're chemically diverse. It just depends on the plant species, which type they'll make. But they all have very strong antimicrobial activity. And they always say they're regulated at the level of transcription. What that means, what they're implying there, is that they're only made in response to a pathogen attack. So if a fungus or bacteria starts to attack, that's when they'll start making the phytoalexins. And they're particularly, they have a really strong ability to make these phytoalexins very, very quickly and make lots of it. And so they'll just ramp up their phytoalexin synthesis, lots of it. So it's concentrated, high doses, and really strong activity. So it's really trying to hammer the microbe right away and hopefully wipe it out. And so it's not something that it does timidly. It's like really ramps it up and goes for it. And so these phytoalexins, when they were first discovered, they were really surprised at how quickly they can get made and how strong they can be against the microbe in terms of killing it. So these are just some different kinds, chemically diverse. It just depends which plant you're looking at and what pathogen might be attacking them. You can see, you know, one group up top, those are isoflavonoids, and then you got sesquiterpenes. So it diverse. We talked about tannins and so forth in red wine, but red wine also has resveratrol. I talked to you a little bit about that, and that's technically a phytoalexin. And technically it's good for us. So that's something that is touted as a healthy thing for our physiology. Not the alcohol in the wine, but the phytoalexin. Okay, so back to this. So you can see how they kind of separated out this box where it says salicylic acid biosynthesis. And then they have an arrow going to systemic acquired resistance. So salicylic acid, that is a hormone, a plant hormone. And so you can see that the calcium and the nitric oxide, it looks like they're ramping up salicylic acid biosynthesis. And then that leads to this phenomenon called systemic acquired resistance. So systemic, what they're implying is that it's a full body response by the plant. And so here they're showing a lower leaf getting attacked by, they say it's an inducer. Maybe it's a fungus starting to attack the tissue there. And then you can see signal transduction. All right, so they're trying to imply that you will have a local response where the pathogen is, but then a systemic response through signal transduction mechanisms. And then you can see another leaf nearby is getting ramped up to defend itself. Okay, so systemic acquired resistance. So this is the idea of, again, we looked at this already with the herbivores chewing on the plant. It's a similar mechanism in the sense that you can have a bacteria or fungus also triggering this systemic response. And what they find is if this plant makes it through this attack and survives, if you experimentally expose them to the exact same pathogen that they just survived, the plant is already ramped up and ready for it. And so this elevated defensiveness is a phenomenon that they didn't think plants had. And so they were very surprised to see that that can happen. And so it's almost like kind of analogous to our memory T cells in our body.
Is a certain time that they degrade the plant tissue, they won't have that elevated defensiveness.
With the fight, what was the phytoalexin part?
Like over time, do the compounds that made it the tissue resistant degrade, so is that resistant? Like, is there a time limit on it?
On the phytoalexin in particular? Yeah, I would think that it wouldn't be permanent. It would be like this burst of phytoalexins. And then it would probably get recycled, like broken down and recycled to get used for other things after the attack is gone. So what is causing the elevated defensiveness? They don't have that nailed down yet. And so they're, and it's not just at the site of attack, it's through the whole plant body. And so they they definitely are looking for the mechanism of that in finer detail. But, you know, like we do that with memory T cells. If you get sick with something, you know, a virus, a cold virus, or well, not really cold virus. That's probably not the best example. But your memory T cells, not all pathogens that attack you will stimulate your memory T cells, but many do. And then when you get exposed again to that same one, your memory T cells are there and they're ready to help you that much quicker. And so that elevated defensiveness. So there's, in order to get this whole systemic process kicked in to high gear, what they're finding is that if it's a fungus or a bacteria, you tend to get salicylic acid hormone produced in response to that type of attack. And then what happens is it gets slightly changed to methyl salicylate. And that version is volatile. And again, that if you think about it, that's not a bad way to go because then they can get that volatile out through their stomata and that can get to distant parts of leaves and branches pretty quickly. And so that's a good option. They also have tracked it through the vascular system, including the phloem. And the parts that go out of the stomata, that portion of it, it can signal distant leaves of the same plant, but it can also trigger nearby plants to start to get up and running. And so we talked about this already with the herbivore idea. It's just that in this case, it's salicylic acid instead of jasmonic acid. So here is they're trying to show a visual of this where here's the pathogen infection. So let's say it's a fungus attacking that leaf. And then they're trying to show that with the red arrows going through the plant that you have salicylic acid, SA, and then the methyl salicylate, they're trying to show the wavy line is trying to show that it's a volatile. And then they're showing a neighboring plant getting that information of the volatile methyl salicylate. And then it gets that plant up and running to defend itself against that exact same pathogen. And so again, this idea of neighboring plants being able to get that signal from the plant that's getting attacked was met with a lot of skepticism, but very careful studies, many studies later the data is really strong. And you can show that this is going in lots of different plants. It's not unique to a certain plant. This is kind of a universal thing. So you can see the two structures of salicylic versus methyl salicylate. So you can see that it, you know, it really does depend if it's a herbivore versus when they say pathogen infection, they're referring to a fungus or a bacteria. And so you'll have different hormones, but some of the same basic schemes of getting it systemic, getting the rest of the plant ramped up, and then neighboring plants as well. Okay, so systemic acquired resistance was the term given for this whole body response to defend itself against bacteria, fungi, viruses. Now there's another term that is also something to think about, and please try to get these two terms straight in your head because they're different. So induced systemic resistance. Okay, so we're still talking about systemic response, but when they say induced, what they're trying to imply is that it's a nearby beneficial microbe that's telling the plant, hey, you got issues, something's attacking you, you've got a bacteria or fungus attacking you, ramp it up. Okay, start defending yourself. So these are beneficial bacteria. They can be biocontrol agents or just beneficial bacteria. There's all kinds of different ones. They're given different acronyms. They can be called plant growth promoting rhizobia or rhizobacteria, plant growth promoting bacteria, plant growth promoting microbes. All right, so there's all kinds of acronyms, but basically, they mostly been looking at, well, they at least started out looking at bacteria's ability to do this, but fungi are also known to do this. So I like the last acronym that's more recent, plant growth promoting microbes, to encompass that it can, yeah, bacteria, but also fungi. And so what's going on here is you have beneficial microbes hanging out with the plant long term. So they're coexisting with each other, maybe even from being on the seed. The microbes might have been on the seed or in the seed. When that plant germinated, started growing, you have that cohort of microbes that hang out. You have a bunch of microbes in the soil that the plant can recruit and develop into its own microbiome. And there's all kinds of interactions going on between the plant and its beneficial microbes. So this is one thing that they've discovered, is that the microbes that are hanging out with the plant can actually signal to the plant, you're in trouble. You are about to get attacked by a pathogen, a pathogenic bacteria or pathogenic fungus. So they found that when this pathway is triggered, salicylic acid isn't part of it. And you don't have those pathogenesis related genes kicking into high gear. Instead, you have jasmonic acid and ethylene. Those two hormones kick in. So this is one, it kind of makes sense that it's a different pathway so that the plant can understand what this signal is coming, who the signal is coming from, that it's beneficial microbes telling them you're in trouble. All right. And so this is the way, because it's a separate pathway, it's a way for the plant to tell the difference that, oh, you're a helpful microbe. You're trying to tell me some helpful things here, so to speak. So they're trying to show that here in this image. So here in the root area, they're showing bacteria, beneficial bacteria. So the way they've drawn this, it almost looks like a root nodule. So they're implying that it might be a nitrogen fixing microbe that's doing this service. Anyways, so what they're showing here, so this is, they're trying to state very clearly, this is a non-pathogenic microbe hanging out long-term with the plant. Here's the plant is infected. Some bacteria or fungus is starting to attack it. And so here you can see the systemic going through the whole plant body. And they're trying to point out that it's these two hormones, jasmonic acid and ethylene. So because it's this different pathway compared to if it's a pathogen trying to attack it, the plant can tell the difference and can ramp up appropriately to defend itself. So when this was first published, this information was met with a lot of skepticism and disbelief, because they just didn't think that a microbe hanging out could alert the plant that you have a bad microbe about to attack or is attacking you. And so again, lots of pushback on this, and lots of studies came after the initial published one. And the data is showing that this is happening. They can very carefully demonstrate with good experimental design that we have these processes going on between the beneficial microbes and the plant that's getting attacked. So here's kind of a big picture idea of all the dynamics, and this isn't complete, but basically they're trying to show that you have all of these beneficial microbes in the soil, on the root, in the plant. You don't forget about the endophytes. They're not really showing the endophytes here, but they also can do this. And so you're seeing that basically you have a range of different microbes, bacteria, and fungi. You have direct antibiotic production against the pathogen. So many of these can produce antibiotics and kill the pathogen outright, which would help the plant, of course. You can see here the plant is essentially recruiting the beneficial microbes by giving the nutrients that those microbes like. It is communicating with these beneficial microbes. So I haven't talked about quorum sensing in this class. I do in my other class, but quorum sensing is basically a way for, in this case, the plant and the microbe to communicate with each other. And they're able to, through that quorum sensing process, they're able to coordinate their behavior in order to accomplish something that they can't do necessarily by themselves. In this case, it's to try to fend off the pathogen. Here you also see the volatiles. They show the volatiles and siderophores. Those are being produced by the microbes that are essentially telling the plant to ramp up. But the siderophores are also helping deal with the plant's ability to get access to iron. So there's a myriad of things going on here that through this beneficial microbe and plant interaction, you're able to get the plant to effectively defend itself against the bad fungi and the bacteria. So this study, this area of study is still very much hot topic because they're trying to understand this a little bit better because they think that the use of beneficial microbes should be something that should be pretty routinely done, especially in growing plants for food or medicinal purposes, that this could really help, especially when so many of the pesticides and herbicides are getting banned and no longer being used. Gotta have other options. And then don't forget about this. This is also something my plants are doing to other plants in this case. So allelopathy, that term is pretty much reserved for one plant producing some kind of molecule that's inhibiting or killing a nearby plant. And most of the time they think that they're doing this so that they can get better access to nutrients in the soil. They can have their fair share and more of the nutrients in the soil. They can have better access to sunlight. This is a study looking at spotted knapweed, which has these pretty purple flowers. It looks really pretty from a distance, but if you see a whole field of that around here, that's not a good thing. All right, this is an invasive in the United States. If you look at the picture on the left in comparison, that's what it looks like over in Europe. You'll see spotted knapweed in a field, but it's a minor component. It's just one of many different species. And so you have a lot of diversity over in Europe where it was native to, and then it got brought over here to America where all of these European species we didn't have. So there was no way to really keep spotted knapweed in check. And it has explosive growth over here where it can overtake all kinds of areas. In that documentary I've asked you to watch on What Plants Talk About, they do discuss spotted knapweed out west and how it's a big problem for cattle ranchers and that it's just getting out of hand. So please watch that. It's an interesting story. So it produces catechin, which is being shown down here. That's the allelopathic chemical, catechin. It secretes that out into the soil, and it's a very potent inhibitor of against many plants. And so it can really start to kill off the native plants and take over. So every summer, there's always job opportunities for summer work where you go and pull spotted knapweed. That's your job all summer long. And you still can barely make a dent in it. It's such an invasive problem. So this is something that's ongoing. The other kind of depressing thing about spotted knapweed is that if you get an area cleared, basically you yank all of it out and destroy it. And then what they try to do is reseed that area with the native plants that should go there, that used to be there, and get it to grow back. And then they find they often don't grow back. And when they went in to ask, what's going on here? Why aren't these native plants, germinating and growing? They realized that the spotted knapweed influenced the mycorrhizal fungi in the soil. And so a lot of the mycorrhizal fungi that hang out with those native plants were destroyed, were gone. And so even though they planted all those native seeds, the mycorrhizal fungi weren't there to help them. And they weren't very successful at getting that area recolonized by the native plants. So it's a long-term problem. It's not just yank out the plant and you're good to go. They're finding that can be many years of effort to try to get that area back to normal. So speaking of nasty plants, here's another one. So here's parasitic plants. So mistletoe is a great example of that. And that's figure A here. Mistletoe is like one of the worst parasites because technically it's called a hemiparasite. Technically, it can live on its own if it wants to. It just doesn't want to. And so it finds parasitizing plants just easier. And so it'll do that. And if you've ever looked up and seen patches of growth like that, mistletoe is more common in the southern states. They actually have people hunt mistletoe. I'm not joking. They actually go out with a rifle and shoot it down. That's the only way to harvest it for mistletoe used around the holiday seasons. I always wondered how they get the mistletoe because they're in dense forests. It's not like you can drag a ladder through the forest. And so they actually hunt the mistletoe. And I listened to a podcast of a father and son team that was part of their business, or the way they made money is they'd go out and shoot down mistletoe. And apparently there's quite a process to it because if you shoot it incorrectly, it just kind of all blows to smithereens. And you want nice clumps. You don't want it all in tiny little bits and pieces because then you can't sell it. And so there's quite an art to it. You have to have a good aim. But anyways, what they're doing is the seeds germinate and grow. There's a special bird called the mistletoe bird that eats the seeds. And then when it tries to defecate out the seed has such a slimy covering that the seed sticks to the butt of the bird. They actually have video. I'll try to post if I can find an active video, YouTube video. They show the bird trying to wipe the seed off its butt on the branch. It's feeding on the tree branch where the mistletoe is. And you can see the bird wiping its butt, and finally the seed comes off the bird's butt, but it's stuck to the branch. And then very quickly, what happens is the seed germinates and its roots start to penetrate into the host branch tree, the tree branch. And so that's showing under the microscope that it's starting to infiltrate the branch tissue. And then it'll start taking away some of the nutrients. So it's green all year. It used to have quite a bit of cultural significance because in the winter, everything else is pretty barren, but mistletoe stays green. And so it was actually a sign of good luck and good fortune. They thought that I think there's some folklore that if people were fighting, like, if there's a war going on and there's mistletoe around, they would try to have a truce. I mean, it had very powerful significance to many different cultures, but really it's a parasitic plant. It's kind of interesting, but here, figure B, that's daughter plant, or no, here, this is daughter. C and D is daughter. And so in that video, you'll also see some information about daughters and how DODDER, daughter, and how it is a pretty amazing parasitic plant. It has a very limited time window to find a suitable host. But then if it does, it can spread like crazy. So there are several different species of parasitic plants, but they all kind of have the same mechanism. Once they attach to their host, they usually have some special tissue that's typically called a hostorium. That's just a general term. It's just a term that they're trying to imply this is a pretty aggressive growth, in this case, into the host plant. And the goal usually is to get to the vascular tissue. and suck away the nutrients. And then there's these kinds of pests. This is an argon tree. This isn't Photoshop. This is for real. They actually do this. I didn't know goats could do this. I mean, I knew goats were pretty amazing on cliffs, sure-footed, but apparently they can climb trees, and they do, going after the argon fruits. So it's the same fruit that does argon oil. It's so good for your hair and skin. So I thought that was Photoshop, but I traced the origin of these pictures, and this is for real. So that poor tree looks like it's pretty much a done deal. Look at that one, way up top. So some trees even have to deal with this. Poor things. So not a whole lot you can do against that type of voracious attack. So a lot of this research that we've been talking about with the production of secondary metabolites and the ability of the plants to defend itself, and then, you know, the idea that one plant can send off chemical signals to nearby plants has raised the idea that maybe there's cooperation going between these plants or among these plants. And, you know, how else are they communicating with each other? And so, yes, there's definitely this volatile bouquet that can get released from one plant and that can get perceived by nearby plants. They're also looking at information going through the mycorrhizal network underground. So the mycorrhizal fungi facilitating the flow of nutrients from plant to plant to plant in even in diverse mixed species ecosystems. You can have the mycorrhizal fungi connecting all of that and the chemical flow through that network. They're looking at ideas like plant intelligence, which is a word or a phrase that a lot of scientists would cringe at. But if you're careful how you define it, there's been whole books written about it and papers written about it. This is a published paper back in 2005. Dr. Trewavas actually wrote a book to plant behavior and intelligence. So a whole book about this idea, and that they're using these secondary metabolites in this process of, when he talks about behavior and intelligence, basically what he's saying is plants are so sophisticated at being able to perceive what's happening around them and to them, and then respond appropriately, that when you look at the idea of behavior and intelligence at a fine detail level, that plants can exhibit this. And so some interesting books and papers out there. Brilliant Green, that's a pretty easy read. Sometimes I find it only for $3 at Amazon. And Dr. Mancuso is, he writes very, you know, no heavy jargon, and he has a very nice writing style. So if you wanted a more entertaining version of something like this, that would be a good one too. This one with Dr. Carbon is a little bit more technical. And then these two are good too. What a Plant Knows by Dr. Kamovitz is also a good one, and it's pretty straightforward, not heavy in jargon. And then Peter Wohlleben isn't a plant physiologist, but he's a forester. He was trained in forestry, and he's written a book, The Hidden Life of Trees. Some of you know about. I've talked about this book in other classes too. But that one has received some pushback, probably more so than any of these others I've listed, because he does use lots of anthropomorphic terms, and that has got a lot of scientists a little bristly. I don't mind it myself because he's writing it to people who aren't scientists. And so there's a goal that he's trying to get this across to non-scientists. So I don't know what the fuss is about really, but anyways, it's a good book. And then somebody asked once, you know, if we're eating all these foods and they're secondary metabolites that are meant to like harm other organisms, how come we're not harmed by it? Or, you know, why is it considered healthy for us? So I did a little bit of looking into that, and I don't have the whole story because I'm not a nutritionist, but you often hear that a lot of these chemicals that plants are using to defend themselves are good for us, like they're anti-cancer or just, you know, good for antioxidants, things like that. And so one example, broccoli, your mom's probably been at you to eat your broccoli your whole life. And so why, why should we eat our broccoli? And it is because the broccoli has secondary metabolites that technically they're toxins. They are used to defend themselves against herbivore or pathogen attack. And when we eat that, what's happening is it really is triggering an adaptive stress response in our body. And it has, the sulforaphane that's in broccoli triggers our stress response. And then it kicks up our own antioxidant enzymes. And so it's not really, when they say eat your broccoli, it has good antioxidants. Well, yeah, it has antioxidants, but the amount in that broccoli is not enough really to be that effective on its own. What it's really doing is stimulating your own antioxidase enzymes. I didn't say that right. Antioxidant enzymes. And so you would have to eat a lot of broccoli to get the amount, for the necessary amount that the broccoli has of the antioxidants, but it's really triggering your own. And so this idea, I don't know if some of you have taken physiology, human or animal phys, maybe this is old news, but I thought that was a pretty cool thing that I was reading. And then you also hear lately the new craze is polyphenols. Polyphenols are really good for us. We need that in our diet. And I agree, everything that I've read about them, I'm convinced as well. But what they're finding is the polyphenols are good for us directly, but they're also helping to benefit our good microbes that are in our gut. And so our gut microbiome uses the polyphenols for their own nutrients. They break down the polyphenols, and some of those breakdown products they give back to us. And they're finding that the presence of the polyphenols increase the good microbes in your gut. And so there's multi reasons why the polyphenols are so good for us. So it's directly affecting our physiology, but it's also impacting the good microbes. And then they will give back to us. So this is taken from that paper where they're trying to show that the polyphenols, we can digest some of them, but some of them we don't. And then the bacteria get it in our guts. And then they act on it and they give us some of the products of that we can then absorb and use into our system. And so they're claiming, antioxidative, anti-inflammatory, anti-hypertensive, and on and on and on. And so I think there is a reason that the polyphenol craze is there. They're finding all kinds of positive benefits. Anyways, okay. No, we're almost out of time, aren't we? Okay, well, let's stop there. So the next topic we're going to get into is stress, abiotic stress. And there's a lot of stresses out there, so I'll have to pick one and maybe one or two and talk to you a little bit about that. So make sure to talk to Duncan if you know there's going to be issues over Thanksgiving break with your experiment. Make sure you talk to him, and then he and I will figure something out